Hi, Histology. Gonna take the first show for you guys here. Let you get a little look at how this is gonna work for the rest of the semester. So today I'm gonna lecture real quick about a few different types of the blood cells, and then, like I said, I'll probably also tape a little something and maybe tack it on to the end of this little lecture with doing some of the digitized slides and actually showing you a little bit more in the real setting like that. So blood is another one of the connective tissues that we've been talking about. This is an electron micrograph of a couple of the different red blood cells there. Again, another one of these connective tissues. It does have fibers. We're going to see that these fibers, in the case of blood, are actually insoluble, and it's only when they're clotting, kind of like hairs getting caught in a drain when they come out of solution, in terms of how they stop blood. It does have the fibers. It does have cells. We're going to be talking about the different types of cells it has, and then it definitely has that background substance. So a lot of times we refer to that as the plasma. So if we're looking at blood, they really talked about the different formed elements of blood. So you have your erythrocytes, which are your red blood cells. You have your leukocytes, which on the list below here are the two different things listed as granulocytes and agranulocytes. Those are your 5 different types of leukocytes, or white blood cells. And then there is what are actually not fully cells, but pieces of cells that come from a much larger cell that you would see in the bone marrow, called platelets. On these things, I'm going to want you to have a basic idea of what they look like, be able to identify them, definitely understand the basic percentages of those normal leukocytes, and again, know a little bit more about just the overall composition of blood. Like I said, I will also, like I said, tape a little bit with the digitized slides to put on the end of this show to actually show you some of these in an actual micrograph as well. So blood, we're going to see, is a connective tissue. It really has those two components, what we refer to as usually the formed elements, which in this slide here we're going to say cells, and the plasma. These different cells or formed elements are going to be the red blood cells or erythrocytes, the white blood cells, also known as leukocytes, and then you have what are called platelets. The plasma is kind of that ground substance of the blood, or the liquid portion of the blood. One of the things you'll notice, it is mainly water. Generally, they say somewhere around 90% or more. And it definitely does have a few things dissolved into it. One of the things I always say about blood is when you're really trying to figure out what is in blood and what is in plasma, because our cells don't swim around, we have a cardiovascular system. So really anything that needs to get into or out of a cell, you're going to see it at some level in the bloodstream. One of the things we see in here are definitely some of these proteins, which are the few things that I think that you maybe should know a little bit about. So the albumins, these are one of the things that keeps too much water from leaving the blood and going into your tissues and giving you edema. These maintain an osmotic gradient, so that idea that water is going to try to follow where dissolved substances are. The fact that these albumins stay in our blood helps us maintain our blood volume and means that we don't get too much of a buildup of fluids in our tissues. Immunoglobulins are another important type of blood protein. These are different antibodies. And then, like I said, fibrinogen is part of that blood clotting process. Fibrinogen is those dissolved proteins that are in solution that when you actually have a break in your blood vessel, that will come out of solution and help along with the platelets to stop your loss of blood. All the other stuff is all those things that need to get in or out of cells. So nutrients, waste products, respiratory gases, sugar, salt, any of those things are going to be part of that other 1%, are all dissolved in there. If you've ever actually seen a blood test, the list is like 2 sheets of like teeny tiny writing of pretty much all these tests they can do on what's in the blood. So one of the things you have to realize is a lot of stuff is traveling within the blood. We're going to really, for our purposes, worry about those things that we can see under the microscope, which are going to be, those formed elements. If you take this plasma and you actually can spin it down really fast in a centrifuge, you can actually get rid of all the plasma proteins in this, and you're left with just pretty much the plasma, which would have the antibodies and everything, minus the clotting ability left, and that's what serum actually is. And this is some of the times that they will actually, so like for example, when they're making antivenoms, they will actually inject small amounts of venom into, let's say, a rabbit or something, and then they will spin down all the stuff there where you're just left with this serum, which would have the antibodies against that particular type of venom as a way to treat that, for example. So let's start out with red blood cells, or the other way you need to know them is erythrocytes. You need to know both names. The erythrocytes, these are the red blood cells. One of the things you'll notice when you look at these, and I'll show you a picture of them in just a little bit, you'll see that they don't have a nucleus. Not having a nucleus is kind of a key thing with these red blood cells. When you don't have a nucleus, it means you can't do any DNA transcription or translation of any of that genetic material, which means these cells aren't going to divide. It also means these cells are not going to have the ability to repair themselves. And when that happens, it really means you have what is, once it's made, it's a cell that is going to have a finite lifespan. In the case of the red blood cells, it's about 120 days, which is really about 4 months. The precursors of all this are derived from stem cells in the bone marrow. These are called hemocytoblasts. And there is this whole differentiation process to honestly get all these different cell types. They all come from that original cell type, the erythrocytes, as well as all the different types of white blood cells. There's a lot of growth factors and other things that regulate that. It is a highly regulated process, but not something in histology that we're going to have to worry about. In terms of the numbers of these ones, it's about 4 to 6 million cells per microliter of blood, or that's 1/1000th of a liter. Basically, a small drop of blood is going to have about 4 to 6 million blood cells. Males tend to be on the higher end of this. Females on a slightly, especially during their childbearing years, are going to be on the lower end of this because of menstrual cycles. Men tend to have the slightly higher numbers, and that would be true in terms of red blood cell numbers, hemoglobin levels, generally anything related to the red blood cells. So when you look at a red blood cell, you can kind of see pictures of them here under the scope. These are quite small. One of the few times we'd actually, if we were doing this in class, would want oil immersion. These are quite small. They're only about 8 micrometers, across. You can see little circular cell. Basically, all you see is red. These ones on a blood slide, if I have you identify them, I'm kind of crazy because when you look at a blood slide, it's basically all you see. You're going to see this and then just a few of the white blood cells and platelets mixed in with this one. But these are what normal red blood cells would look like. Like I said, they're a biconcave disc. So if you think about like a donut, but having the inside of the donut filled in, that's what we get by a biconcave disc. This shape gives them a couple of benefits. One of these is that when you have a spherical cell, if you are going through a narrow area and the cell's a little bit bigger, it's going to try to go through and get stuck. By having this biconcave disc, they have this ability to flex a little bit. The other thing it does is we need oxygen and carbon dioxide to be able to diffuse into these cells and actually travel, and a biconcave disc is one of the ways we can actually increase our surface area for the amount of volume that's in there. So this biconcave disc is giving us a lot of surface area for not as much cellular volume. Again, mainly water. There's going to have very little in the way of organelles, and we have a lot of this protein called hemoglobin, which is the oxygen carrying protein within the cells that is going to make up the main part of that. One of the things you'll see that, well, you won't see, but one of the things that you need to know is that there is actually no mitochondria, for example, in a red blood cell. And this is mainly because the mitochondria are going to use oxygen to generate use oxygen to generate ATP, and by the fact that the red blood cells are here to carry oxygen, you don't want to be like the pizza man eating up all your pizza instead of delivering it. So we don't want our red blood cells using up the oxygen that they're supposed to be carrying. So because of this, we don't have organelles that allow them to generate energy that much. They're doing basically just simple glycolysis for any of the ATP needs they need. And this is, you can kind of see at the bottom of the slide here, you can see that it is that biconcave disc, again, giving us a lot of surface area for not a lot of volume. And again, as I was saying, that shape allows them to bend and twist a little bit more. As these get older, that ability is lost, and that's part of the reason that they end up bursting as they get to the end of their lifespan. They lose some of that flexibility as they age, kind of like humans do. To kind of show you hemoglobin, this is a picture of hemoglobin here. I think you can see my cursor here. You can see it has 4 chains. In the middle of each of these globin chains, you're going to have what's called this heme group. In the middle of that heme group, I want you to kind of look at this molecule right here. This Fe stands for iron. We need an iron molecule in the active site of each hemoglobin molecule in order to be able to carry an oxygen. So what we end up seeing is on each hemoglobin molecule, it can carry 4 molecules of oxygen, and this is what is going to allow us to carry on that oxygen in our bloodstream. The other cell type I want to talk to you about real quick is the platelets. So the platelets are made from these cells called megakaryocytes. Sometimes platelets are referred to as thrombocytes. These actually are not fully cells, but they're actually just pieces of cells. And like I said, they come from those megakaryocytes that are in the bone marrow. These are large cells that are actually a combination of a bunch of cells fusing together, multinucleated, that they come from here. We have about 150 to about 400,000 of these per microliter or per small drop of blood. Quite tiny. When you look at them under the scope, they actually look like little purple specks of dust. Again, no nucleus, going to have a few hormones and a few other substances in them, but are basically little packets of cytoplasm. And again, their job, I think as most people are aware, is to be involved in the stopping of blood loss. A lot of times that's referred to as hemostasis. They can trigger a clotting pathway and, again, help us if we do have a break in our blood vessel from losing that blood. If you look at the picture right here, you can see what the arrows are pointing at. All those little purple specks on the screen here are these small little platelets. The large cell in the middle is actually a leukocyte, the large purple cell in the center, which we'll talk about those in the next little slideshow I'll do here. But you can see where these arrows are at right here. You can see these platelets out and about in here, again, looking like little specks of purple dust. Again, this is a micrograph of the large cell that they, this is actually a micrograph of the platelets. They have some small looking granules in them and a few receptors on the outside. These allow these to actually stick to breaks in the blood vessel a lot of times when collagen is exposed and trigger some of the clotting cascade and some of the other stuff that's going to be involved in stopping blood loss. And again, they can stick to these breaks in here, and you can see on the second picture on the right here where all these platelets have stuck to this and helped to try to seal up this opening. And that's it for this first one here. Like I said, when I come back on the next one, we will talk a little bit more about the different leukocytes and how we can tell those apart. So real quickly, I want to take a look at one of these slides. So this is one of the ones from the University of Michigan. I was going to take a little zoom in on this and show you a little bit about the red blood cells and the platelets that are on here. And then, like I said, on a subsequent show here, we'll take a look at some of the actual leukocytes. Those are the ones that are a little bit more difficult. So what I'm going to do real quick is kind of zoom in on this a bit. And as we're getting in here, you can see generally on a blood smear, you can kind of see all over the place. There's a lot of these red blood cells. Those, like I said, are quite easy to spot and quite easy to deal with. We're generally going to need to zoom in a little bit more. And then if you start looking around on some of this, you can see kind of where the pointer is right here now, a couple of those platelets here and there. And honestly, on that one, and again, another platelet here, you can see red blood cells about everywhere. As we get into this a little bit more, like I said, I will start showing you, you can start seeing, so that cell right there would be one of the different types of white blood cells, and we'll get into those on the next one. But as you can kind of see, at least identifying red blood cells and platelets is pretty straightforward. So again, we'll pick up with this next time with the different leukocytes.